Hello. Welcome to the next in a series of review clips designed to assist you in preparing for your Biology 3201 public examination. In this clip, we're going to examine the concept of cloning. Cloning is, by definition, the production of an organism that is genetically identical to one another. In short, organisms that are genetically identical are clones. Asexual reproduction always produces clones, and there have been laboratory techniques developed that have allowed this to happen in animals. In the 1950s, the first experiments were indeed done with frogs. And what happened is transplanted nuclei from cells of tadpoles and frog embryos were transplanted egg cells that had had their egg nuclei previously removed. This was done by Briggs and King. What they found was that many of the eggs would develop into tadpoles if the source of the original nucleus was the early embryo. When they took tadpole nuclei, they would not. They also found that the tadpoles that did arise would not develop into adults. These results gave support to the idea that differentiated cells could not be used to create clones. In the early 1990s, however, there were some success stories found with mice. Mice were cloned by using nuclei of cells taken from mice embryos. And in 1997, Dolly, the sheep, was produced. What they did, Wilmot and his colleagues, was they used genetic information taken from the udder of an adult sheep. Now, in order for differentiated cells to be cultured to produce an undifferentiated embryo, the process of cell differentiation had to be reversed. To do this, what they did was they took an egg donor. Again, unfertilized eggs were collected, their nuclei were removed and discarded. At the same time, from the udder of another sheep, a separate sheep, these udder cells were collected and treated to prevent cell division. They in turn had their nuclei removed. The nucleus from the udder was transplanted into the empty eggshell. To restart the cell cycle, an electric current was applied. Some of these cells divided to produce an embryo. This embryo was implanted into the uterus of a surrogate sheep, and the lamb that was born as a result was identical to the donor of the udder nuclei. This sheep and the lamb were genetically identical. In short, they were clones. So, just to outline the process again, a donor cell was taken from a sheep's udder. At the same time, an egg cell from another sheep was removed. The nucleus of the egg cell was removed. The donor nucleus was implanted into the cell that was empty. These cells were fused using an electric shock to create an embryo. These cells started dividing to form a normal embryo, which was in turn implanted into a foster mother. The lamb, Dolly, was genetically identical to the original sheep which had donated the udder cell. They were clones of each other. There have been problems with this research. Other research have indeed repeated the work, but as more examples are created, there is evidence that there are health problems. Dolly showed signs of premature aging and died much younger than a normal sheep would die. There are some theories that suggest this is related to the shortening of the telomeres with adult cells. With regards to human cloning, there is no disputing this is a very controversial topic. But we have to draw a distinction between therapeutic cloning and reproductive cloning. Therapeutic cloning is the culturing of human cells for use in treating medical disorders, whereas reproductive cloning is developing a cloned human embryo for the purpose of creating a cloned human being. There is certainly a marked difference between the two. Those that are in favor of therapeutic cloning argue that this may provide the key to eliminating disease. Yet, we cannot escape the reality that all means of cloning does result in both the creation and destruction of hundreds of embryos. Cloning technologies certainly have the potential to change how we define life. Past public examinations have asked questions on this topic, and I present the following. Which technique could allow doctors to create perfectly matched bone marrow for a cancer patient? Differentiation is simply making different. Again, this would not be perfectly matched. Gene therapy would be injecting genes that were deficient. Reproductive cloning involves reproduction, not to create medical treatments. The correct answer is D, therapeutic cloning. Constructive response question has been posed as well, dealing with the ethics. This question is posed. Explain either two reasons for supporting or two reasons for opposing the use of therapeutic cloning to replace damaged cells in the spinal cord. The acceptable answers: 4, the ability for the person to lead a normal life. Nerve cells grow slow, so may need cloning to improve their rate. It may lead to future discoveries in spinal research, and it doesn't have to use embryos. Those in opposition, arguments for it: the idea of playing God, religious convictions, are you killing embryos, which is certainly a possibility, and certainly the idea of rejection by the new person. 1 mark was given for each of these answers. Another sample question is posed. Which is a potential benefit of cloning? Embryo use and destruction would certainly not be a benefit. Loss of individuality, again, not a benefit. Nor would reducing genetic variability. The correct answer would be A, elimination of disease. This question is posed, asks, which involves culturing human cells for treating medical disorders? The key word to focus on is treating medical disorders and culturing human cells. Amniocentesis draws cells, but you are not treating, you are diagnosing. Cystoscopy involves looking at the fetus as it develops. In vitro maturation certainly plays a role. The correct answer, however, is D, therapeutic cloning. Thank you for your time. I hope you found this learning clip to be helpful.